Hey guys, Mr. P. In this video we're going to talk about the cell cycle, specifically D2113, which is phases of the cell cycle. So the understanding states that students should understand that cell proliferation is achieved using the cell cycle. Students should understand that sequence of events including G1, S, and G2 as the stages of interphase, and that is followed by mitosis, which is then followed by cytokinesis. So this specific video is not a lot of content related to what happens within the phases. This is more of an overview of the cell cycle in general. It also is going to highlight the sequence of events from interphase, mitosis, and cytokinesis. So the first thing we need to do when talking about the cell cycle is obviously look at the sequence of events. Interphase consists of G1 through S, and then ultimately through G2. So if you look at this little blue inside area of the diagram, you can see that it is labeled interphase. Interphase consists of, it starts here at the end of M phase, specifically at the end of cytokinesis, and will run through the normal cell activities, the cell functioning, from G1, S, and G2. So interphase consists of G1, S, and G2. Also notice that off of G1, at the beginning of G1, there is a stage called G0, and G0 is a state of stasis. Cells go into or can enter G0 when they want to remain static or when they want to remain homeostatic for a while. Some cells will enter G0 and never leave, and therefore will never be able to replicate themselves, like brain cells. Some cells will enter G0 for a little duration or a short duration while they maintain stasis, and then will exit G0 and then continue through their normal growth and then proceed through normal interphase. G1 is the first phase of interphase, and in G1, cells accomplish most of their growth. They get bigger in size, make proteins and organelles needed for normal functioning and expression of genes. So a cell will start small after it divides and will exit G1 or enter S as a larger physical cell than when it started. All of the cell growth happens during G1, or I should say most of the growth happens during G1. G stands for gap 1. It does not stand for growth, even though growth does start with a G and growth occurs in G1. G1 starts for gap 1. It is the first gap of the particular cell cycle. So the RNAs, the centromere specifically, and other components necessary to produce the centrosome are also made in G1. That's going to be important because these components are going to help aid in the division or splitting of the cell nucleus, which are going to take place in the cell division part, which is M phase. S is called DNA replication. S stands for synthesis phase. S follows G1, and in synthesis phase, the cell is going to synthesize a new DNA molecule that is called DNA replication. We'll have actual in-depth videos about the intricacies of DNA replication later. But DNA replication is the process used to make additional copies of the cell nuclei DNA, and it has to be replicated in order for the cell to divide not only its nucleus but its cell, but its cytoplasm later on in the cell cycle. Two identical DNA strands are attached to each other at the centromere, and so when a chromosome replicates itself, it will start out as an unduplicated chromosome, and it will replicate itself to produce the duplicated cell structure, which is made up of two identical sister chromatids, which are held together by that physical structure in the middle called the centromere. In G2, again, G2 is the third phase of interphase. G2 stands for gap 2. It's going to follow the S phase, and in gap 2, this is all about the preparation for mitosis. So the cell is going to synthesize proteins needed for cell division. It's going to break down the cytoskeleton, which is the physical structural components that make up not only the size and shape of the cell, but also provides a lot of transport properties within the cell. They are the components that are also holding organelles in place, and it is growing in size by increasing the cytoplasm volume It is a G phase, so there is additional growth. Even though the majority of the cell growth happens in G1, there's an additional bit of growth that happens in G2, but basically the preparation for M phase is going to take place here. So when we kind of look at interphase as a whole, interphase starts with G1. The cell is going to physically grow and get bigger. It's going to replicate all the organelles. It's going to do all of the normal functioning. It's going to express its genes. It's going to produce RNA and the centromere and centrosomes. It's going to then enter S phase, which means it's going to replicate its DNA and produce identical copies that it can then divide into the 2 new daughter cells. It enters G2, which is preparation for mitosis, and there it prepares all of the proteins and necessary components for the proper division of the nucleus. We're going to enter after interphase M phase. M stands for mitosis. Mitosis is the cell division process. M phase is actually going to not only contain the 4 phases of mitosis, notice prophase, metaphase, anaphase, and telophase would be the 4 phases or subphases that make up mitosis, but M phase also contains cytokinesis, which is a separate standalone process contained within M phase, and cytokinesis is all about cell division, or the part of cell division that is specifically splitting the cytoplasm. M phase, on contrast, is part of cell division in which the nucleus divides. So if you talk about what is cell division as a whole is made up of mitosis and cytokinesis. So M phase is cytokinesis and mitosis. Mitosis divides the nucleus. Cytoplasm is divided via cytokinesis. So what do we use M phase for, or what is the big deal about cell division? Well, we just talked in our previous video about the functions of cell division, which is all about tissue repair. Organisms need to be able to replace and repair dead or damaged tissue cells. Organisms also have to have the ability to grow. So growth occurs at the organism level because cells are being produced, and the organism is specifically getting more cells that it is composed of, meaning it will get bigger. Embryonic and fetal development, replacement of cells that die naturally, clonal selection of B cells that is related to immunity, we'll talk about that later. Asexual reproduction via cloning. Some cells have the ability to reproduce via cloning, and that is a form of asexual reproduction. Maximizing the cell surface area to volume ratio, what does that mean? Cells are actually more efficient if they are smaller. Smaller cells are more efficient because they have a higher surface area to volume ratio. As a cell gets bigger, its surface area to volume ratio decreases because the volume is increasing faster than the surface area. And that'll do it for this particular video. If you learned something, give it a thumbs up, subscribe to the channel. We'll see you later.